Hello and welcome everyone. This is our second podcast on eukaryotic gene regulation. In this podcast, I'm going to focus on how eukaryotic genes are regulated at the transcription level, but focus primarily on histones and how they are removed or added to affect transcription, and then transcription factors and their role in regulating gene expression. Okay, let's go ahead and get started, and we're going to start with transcription factors. So let's write transcription factors up here. We're going to talk about two broad classes of transcription factors. One we've already mentioned, and those are the general transcription factors. And then the other one, I briefly mentioned them before, but we didn't spend any time talking about them to any great extent. And these are the regulatory transcription factors. Now, the term transcription factor is a term that's often used to describe proteins or enzymes that somehow or another influence whether or not a gene is transcribed or not transcribed. We're going to focus now these factors could be regulate various parts of the transcription process. We're going to focus on just that early part of recognizing the promoter and initiating transcription. Once the genes start to be transcribed, there's not a lot of regulation that occurs, at least not that we know of, in the elongation or the termination stage. So we're going to focus on that early part. Okay, so let's talk a little bit about the importance of transcription factors. Now, obviously they're important, but let's put that into some perspective. In plants, about 1,500 of their genes are transcription factors. If I put TF, I mean transcription factor. Humans, about 2,000 of our genes are transcription factors. And as a point of clarification, when I said plants, I'm talking about a specific plant, Arabidopsis. This is a very common model organism for genetics research. So you can see a very large part of our genomes and the plant's genomes are devoted to making these transcription factors. And these are thus very important, as you can imagine, because if you don't have these transcription factors available, or the right ones available, you don't turn on the genes that you need to have turned on. We won't have a lot of time to talk about it, but transcription factors have a very interesting evolutionary history. They are also linked to many kinds of diseases. Cancer, just to name one of them, several types of cancers are linked to various transcription factors. And because of that, often transcription factors turn out to be a target in drug discovery and drug design. But let's go ahead and focus on these general transcription factors and regulatory transcription factors for right now. Now remember, we talked about these general transcription factors before. Let's go ahead and write our gene here, our eukaryotic gene. So we'll have our open reading frame here with our transcription start site here. We'll have our -25 region here, which is that ta-ta box. And so this ta-ta box can be within this -25 region, and the placement of it can have a large impact on the type of, or the level of transcription that comes from this ORF. Also, even though it is a consensus sequence, this area can tolerate small changes. You're not going to make it GGCCC and expect it to work. No, that wouldn't work. But you could expect like a TAT, TA, something like that could still work. Typically, you're going to see a lot of T's and A's in this area, so it's a place where the DNA can be pulled apart. And it is this area here where our general transcription factors will bind. Remember, we talked about TF2. Again, that stands for transcription factor for RNA polymerase II, D. And that it contained many proteins in it, but the one that was of particular interest was TBP for TATA binding protein. And then we will also have areas upstream, and we said they might be upwards of 50 to 100 base pairs or even further away, but sometimes they can be right next to the promoter or close to it. And these we'll give a kind of a general term with, we'll call them regulatory sequences. They allow for a higher level of control over the transcription that happens from the promoter. Now remember, we mentioned before that the level of transcription that occurs just with these general transcription factors is enough to get a low-level transcription. Depending on what kind of regulatory sequence this is, if it is a silencer sequence, it will bind a repressor protein. So a repressor protein might bind here and interact with this area or the transcription factors here in such a way that it eliminates transcription. So again, these by themselves allow for that low-level expression, but the proteins that bind to these regulatory sequences can dramatically up or down-regulate this gene. I should probably define those two terms I just used, up or down-regulated. Repressors will down-regulate the gene. They will turn it off. So down-regulate. They turn the gene off or they reduce the expression. These general transcription factors are trying to turn it on. They do to some limited excess, but then a repressor binds to a silencer sequence and it turns off. Next, I want to talk about the other kind of sequence we may have here. So I'm going to erase our silencer sequence and the repressor protein and talk about another class of sequences. And so we could have a regulatory sequence that is known as an enhancer sequence, and we mentioned this before. And when the enhancer sequence binds here, it can interact with the general transcription factors here. And remember, the general transcription factors were giving us this kind of low-level expression. But when a protein binds to this enhancer sequence, and of course it's not just any protein, it would be an activator as a general term to call these proteins that do that. And they upregulate the expression. They turn it on at a faster rate. So to quickly review these two terms, transcription factors include TF2D and the TATA binding protein, which initiates all the other transcription factors coming in here, as well as RNA polymerase. That is enough to turn this expression on at a very low level. To fine-tune that expression, we have these regulatory sequences. They can bind enhancers, as I have drawn here. When at these enhancer sequences, when an activator protein binds to them, it will affect the general transcription factors in the core promoter here and drive this reaction to a faster rate. Alternatively, and I've already erased it, but we also have silencer sequences here. And you can have both sequences right next to each other here. The silencer sequences, they bind repressors, and those repressors, remember, will turn this expression off. Okay, let me go ahead and erase this. Okay, let's talk about in a greater detail how these regulatory transcription factors affect transcription factor 2D from binding to the promoter. So let's draw our famous gene here, and let's draw our core promoter here, the -25 region, and then let's draw our regulatory sequence here. Remember, these regulatory sequences could either be enhancers or silencers. And we know that the transcription factor 2D here, the collection of proteins that are all this factor, wants to bind here and begin to allow transcription to occur. But we said that these regulatory elements here play a role in whether or not how it's going to affect that. So let's first talk about repressors. Repressors are going to bind to this element here, and in binding to it, they act in a silencing way. because they will either bind directly to the transcription factor 2D protein, preventing it to bind to the promoter itself. Alternatively, this repressor could cover this -25 region, preventing the transcription factor 2 from binding. Either way, it's blocking TF2D from doing its job. This is TF2D, remember. The way you remove this function is, or you can imagine if once the repressor is removed, let's go ahead and remove it then. It's removed by some factor, and now this allows our transcription factor 2D to bind to this promoter. And once it's bound, it can now allow transcription to occur. Now sometimes, let's go ahead and erase repressors and silencers, because now we want to talk about enhancers. This transcription factor 2D, of course, is going to do a good job at getting this low level of expression from this gene. But if we wanted to stabilize it so that it was more firmly attached to this promoter so it could constantly attract the other transcription factors and the RNA polymerase, you could imagine a protein that could bind here. So this is going to be an activator protein. And this activator protein is binding to this enhancer sequence. And it's going to cause an upregulation. And how is it going to cause an upregulation? Typically how it works is it could directly bind to it, but for whatever reason, there's usually some additional protein that serves as a link. And we call this additional protein, let's see if I have room up here, it's called a co-activator, because it links the true activator here with the transcription factor 2D. And when it's bound to the transcription factor 2D, it stabilizes its interaction to the promoter. By doing so, transcription factor 2D is not going to fall off the promoter. Something we really haven't talked too much about is that transcription factors, or really almost any DNA binding protein, when it binds to a DNA, or in this case binds to the promoter, it binds in kind of a leaky way, meaning that it binds and it falls off. It binds and it falls off. But this activator stably holds it onto the promoter so it doesn't go on and off. In doing so, then what happens is we increase this expression multifold. Okay, I want to next talk about these mediator proteins. And I don't want to say much about these. Your book goes into a little bit more detail than I think we need to know at this stage. But if you remember, I did mention a while back that sometimes these enhancer sequences, or the silencer sequences, can be very far away from the promoter. So if the promoter is here, and the enhancer sequence or silencer sequence is here, we mentioned how can they influence each other. So remember, this was our core promoter, our -25 sequence, the core promoter. And the question was, how does this sequence influence here? And at the time I said it can bind here and interact with any of the proteins that happen to be here. So it could be either the basal transcription factors, or it might be the RNA polymerase. So let me write RNA polymerase. I call them basal, but I should call them general transcription factors. Often when this enhancer sequence is far away, even though they can interact because of the bending of the chromosome, often still that does not put them in the proximity that they need to be. And that's when these mediator proteins can come into play. And a mediator protein, as the name sort of implies, it mediates the interaction between the silencer, the repressor or the activator protein here, and either the RNA polymerase or these general transcription factors by binding in between. Let me erase GTP here. I don't want you to think that this is a GTP. So let's put GTP here. General transcription factors. And then this is our mediator. As I said, your book goes into a little bit more detail about how it influences these, but all I really want you to know is that sometimes these proteins, in order to facilitate their interaction, they require this additional protein. And it can be additional several proteins here. But this general class of proteins that mediates the interaction between a silencer or a repressor and the enzymes necessary for this basal transcription, these general transcription factors, and the RNA polymerase. They use these mediator proteins. That's all you need to know. So up to this point, we've talked about the various enzymes and proteins that are necessary to regulate transcription. But when you're talking about a full organism, so we'll just draw a stick figure here, how does this organism, this human here, tell one part of the body to turn expression of genes on a different part of the body? That is, how do we actually cause this regulation to occur? What kind of signals have to happen? How does a particular cell know to turn on this? And one way that we can do this is by using hormones. This turns out to be a very important way to regulate which genes get turned on or off. So let's just take a random cell from this individual here. Hope he or she does not mind. And here's the blown up version of that cell. And your book gives a specific example of glucocorticoid. But let's just talk about this in a very general sense. Just think about it as any given hormone. And we'll draw it as kind of this triangle. This hormone is taken up by certain cells that need to turn on certain genes. So it's brought into this cell. One way that transcription factors are regulated is by regulating whether or not the transcription factor is in the cytoplasm or the nucleus. So let's draw the nucleus here. Our good friend, the nucleus. And now let's draw a transcription factor. And let's say for some reason that the only way this transcription factor can work and get into the nucleus is if two of them bind together. But for whatever reason, they're kept apart in the cytoplasm. It could be that they have some kind of charge on them that prevents them from interacting. There could be some sort of amino acid that is sticking out of them that needs to be taken up, cleaved off. But for whatever reason, they are not interacting. And so they can never make it into the nucleus in order to do their job. Well, what the hormone will do is it will come along here and interact with these two subunits of this transcription factor, causing them to come together. And when they do, they're now able to enter into the nucleus. And once in the nucleus, they can begin to transcribe a gene or to regulate that gene. So here's our gene. We'll put our promoter here. And of course we have a regulatory sequence here, which may be enhancer or silencer sequence. And now it can exert its effect upon these genes. And let's just say it's to activate these genes. And so now, because this hormone entered into the cell, because somehow the body told this part of the organism that we needed to express this gene. So now, once inside the cell, the hormone brings these two transcription factors together. They now enter into the nucleus, where they can bind to the promoter and cause the gene to be expressed. So you can imagine all kinds of varieties of ways this could work to turn on the right genes. Let's say you've just eaten a cheeseburger, and now your body needs to metabolize that. And so in order to metabolize that, there's some endocrine gland in your body that releases this hormone. It then enters into the cells, say, of your digestive system, and then turns on this transcription factor, which then turns on genes necessary to produce enzymes that are important in digesting certain components of that cheeseburger you just ate, or whatever it is that you might have eaten. Okay, let's go ahead and move on to the next topic, and that topic is histones and how they are modified to affect gene expression. So next we're going to talk about chromatin remodeling. So remember, we may have this long stretch of DNA, and we may have a promoter here, and we may have transcription factors wanting to bind to it, but if this DNA has histones around it, then there's no way the transcription factors or the RNA polymerase can have access to it. So, as I said on the last whiteboard, if that promoter is buried in a nucleosome, it is not accessible to all those proteins we just talked about that can activate transcription or repress transcription. So we could think of DNA, and I'm going to draw the DNA like this right now. And the reason I have it all packaged up like this is because we have our nucleosomes attached in. I'm only showing one half of them. Remember, the nucleosomes contain 8 histone proteins, and the DNA wraps around these histones, protecting them, and also aids in being able to package up the DNA, as we've talked about before. So what happens if we have a gene, say right here? All of our favorite transcription factors, or RNA polymerases, or activators, or repressors, all of these things we just talked about, every time they try to access this region here, they're blocked. They can't get to it. This gene and its promoter is hidden. So how do we remove these histones? I'm going to erase our proteins down here. It turns out that these histones are by and large fairly positively charged, particularly the areas that interact with the DNA. And remember, DNA is fairly negatively charged. And it's negatively charged, in fact, why don't we just write that here in the same blue color just so we remember that. It's negatively charged because of the phosphate group. And so this makes for a nice ionic interaction between the nucleosomes and the DNA. So if there was some way we could remove this positive charge, or at least cover it up so it's not as positive, that would weaken this association between the histone proteins of the nucleosome and DNA. And so we have this enzyme called, I know another enzyme we have to remember, there's a lot of enzymes. It's called histone acetyltransferase. We often just call it HAT for short. And so that's what we'll do. What this HAT enzyme does is, as it says, it adds an acetyl group to a histone. It's going to add this acetyl group right here at these positive charges. But only in cases where we want to turn this gene on, when we want to activate this gene. Once that's happened, the DNA can relax and is not as tightly associated with the histones. So I'm going to erase the middle part here to show you the histones being removed and the DNA coming loosening up here. So the histones have now been released, and they still remain closely associated with the DNA, but they have this acetyl group on them. So they don't just disintegrate and go away. They're around here. Though I'm not drawing it that way, these histones are still loosely associated with the DNA, but it does allow the DNA to kind of relax a little bit, like so. And now what we've done is we have exposed that gene we wanted to activate. And this gene now will be accessible to the transcription factors. Well, I'm going to erase this histone group here. It's going to be accessible to the transcription factors, both the general transcription factors and the regulatory transcription factors. RNA polymerase will now have access to it. And so now that these histones have been removed, the chromatin has been remodeled, exposing the DNA, allowing the genes, the enzymes necessary to activate the gene to have access to the gene. Now, what if we wanted to turn this gene off? Well, what happens is we have to remove these acetyl groups. So there's an enzyme that's going to come along here, remove the acetyl groups. When the acetyl groups are removed, and this is why these chromatins are always closely associated with the DNA. Once it's been removed, it will now bind the DNA tighter. And let's draw that again. Acetyl groups have been removed, so now our histones once again tightly bind up the chromatin, and again hiding our gene here so it cannot be expressed. And the name of that enzyme is called histone deacetylase. So it's a name that makes sense, right? Because it removes the acetyl group from the histone. And now the histone is once again positively charged, so it can bind to the negatively charged DNA. And this turns off the expression of a gene. So we haven't talked about cancer yet. We will. But to give you a bit of a preview here, cancer is caused by, essentially, uncontrolled growth. So we could maybe think about how a gene that's regulated by these histone acetylases and histone deacetylases could lead to uncontrolled growth. So let's pretend then that this gene here that we've been talking about is a gene that codes for a protein that causes cell growth. So when we want the cell to grow, we want that gene expressed. We want the histones to come off, and so it can be expressed. But when we don't want cell growth, maybe there's a mutation, or maybe it's just not time for that cell to divide yet, we want the histones on. So in a disease like cancer, which is characterized by uncontrolled growth, what kind of mutation in these cancer cells would allow this gene to be permanently expressed? That is, the mutation would allow these histones to not be associated with it, so that the gene is permanently turned on, so that the cell will always grow. So think about that. Would we want to have, would the cancer cells, I should say, have a mutated histone deacetylase, a mutated histone acetyltransferase, a mutated histone, something else? Think about why the histones are binding to the DNA. They're binding to the DNA because the histones are positively charged and the DNA is negatively charged. The histone acetyltransferases add those acetyl groups to make these histones fall off, and the histone deacetylases remove that acetyl group so the histones can come back on. Can you imagine a situation where either one of these could be mutated to cause cancer, or the histone to be mutated to cause cancer? Think about that. That's a good way to kind of review this, and we'll talk about this in class some. That's all we have for this podcast, so let's close with a summary. In this podcast, we've talked about quite a bit of things here, but in general we focused on how a gene is regulated at the level of transcription factors. We talked about general transcription factors and regulatory transcription factors, how the general transcription factors can exert a basal level of expression from a gene, and then how the regulatory transcription factors can exert a stronger influence on that gene by either shutting it down completely or by fully activating it, by upregulating it or downregulating it. We then spent quite a bit of time talking about transcription factor 2D, that binds, which is a general transcription factor that binds to that core promoter, and then how the regulatory transcription factors influences the ability of TF2D to do its job. And so we talked about repressors that bind to silencing sequences, and when they do that, they interact with TF2D in a variety of ways, but the end result is that it down regulates that gene. Then we talked about activators that bind to enhancing sequences, and when they are involved, they will interact with transcription factor 2D here and fully upregulate that particular gene. We spent a little bit of time, but not a lot of time, talking about mediators and how they can just help mediate this interaction between a repressor or an activator with the transcription factor. Again, we didn't spend nearly as much time as the book spends on this topic. We just talked a little bit about how they help in this interaction between these two sets of regulatory transcription factors. And then we closed with talking about hormones to kind of give a reason why we're doing this, or in this multicellular organism, how do we tell certain cells to turn on genes and not tell other cells to turn on genes? And we talk about how hormones can do this by entering into a specific cells, and interacting with transcription factors in the cytoplasm and bring them into the nucleus to do their job. Now that was just one example. Hormones can enter a cell and activate transcription factors in other ways, but this was just one particular example. Alright, that's all I have for now. If you have any questions at all, please make sure you come talk to me. I'll see you in class. Bye.